quick one, remind you about the morphological unknown. If you went to the website anytime between Thursday and tonight, you would have noticed you have 3 assignments up there. And some of you might have just gone, what? 3 assignments? Well, so I would like them to be turned in by September 28th. That gives you some time to work on them. One is the morphological unknown. Well, some people were asking questions about the morphological unknown last time, and I thought, wow, are you reading the document? So your morphological unknown, you have a report form. And on that report form, you have sections that you're supposed to fill in. The first, right at the top, that's for me to fill in. Questions like, is the culture pure? Are the colonies well isolated? Is it correctly labeled? I'm going to grade that. Don't fill it in. But if you want to fill it in, go ahead. But I'll write over it because I'm going to grade that. So that's that little top section. And then you need to know the difference between colonies and cells. This comes into play in a couple of places because you have one section where it's asking you about colony morphology. Which of those can you see with the naked eye? Colonies. These are visible on plates, specifically Petri plates. The other thing I found out is people don't know the difference between a plate and a glass slide. Oh my word, plates. Auger plates. That's what these are. Plates. What's on here is colonies. So that whole section that is talking about colony morphology, that's where this comes from. And yes, you have to measure the colonies. Colony size is quite variable. So if you're measuring colony size, typically you look for the largest. And let that be your guide. Use that for your descriptions. You're looking at form, margin, elevation, surface texture, optical character, etc. We measure them. The size is in millimeters. Okay? So you have a set of boxes you're supposed to fill in. There's also a question in there about, does your colony produce any specialized, something that you can see? Some of them do produce crystals, but not very many. We have one morphological unknown that is supposed to be fluorescent. If you have that one and you would like to see the fluorescence, let me know because we have to put a UV light on it to see the fluorescence. We don't have any bioluminescent ones out for that unknown. So then you have cell morphology. Well, first of all, you have size, shape, and arrangement. To get the most accurate size, and this is in microns, micrometers, to get the most accurate size, use your indirect stain. Some of you did those from the broth tubes initially. If you didn't get good results from the broth, just go back and do it again. This is a really quick preparation. Cells in water, a little loopful of nitrogen, spread it out, let it dry. The reason we use the indirect for size is because the cells are fixed, that is, the stain is fixed without heating or exposing the cells to any kind of chemical. They're just in water. The nitrogen solution is aqueous. You let them dry, you get a good indication of size, also shape and arrangement. For size, if they are cocci, you're just measuring diameter. But if they are bacilli, you have to measure diameter and length. If they are spirilla, we only have one of these, measure length without trying to figure out what they would be if you stretched them out. You know, like a spring, you don't do that to them, just measure length that way. But again, you have to measure diameter. And you should measure more than one cell. Think about if somebody asked you, how big are humans? We're not all the same. Big ones and little ones, fat ones and skinny ones. Measure more than one. So what you're going to list for size is a range, size range. You're going to have more than one. Now, a lot of them, many of the cultures, the diameter could be consistent throughout, but the length, if they're bacilli, the length is going to vary. And on most of the cocci, you don't just have one diameter. You'll have more than, some are little, some are bigger. The only problem we run into is the smallest unit that you actually have to measure is 1 micron. And very often the cells are a little bigger than that or a little smaller than that. So you have to estimate, say, well, it's 1.1 or 1.2, or it's 0.8 or 0.9. Anyway, so that's cell size. And then you have the stains. You always have data, result, and conclusion. This is pretty straightforward if you're talking gram stain. Data is purple versus pink. Result is positive versus negative. Conclusion is, what does that tell you about the wall? If the cells are pink, they're gram negative. That means their wall is thin peptidoglycan with an outer membrane. If they are purple, that's positive. They are thick peptidoglycan, no outer membrane. If you're doing an acid-fast stain, for some reason, acid-fast is not recorded, the result is not recorded as positive negative. They're either acid-fast or they're not acid-fast. But again, it tells you about their wall. If they're acid-fast, they have mycolic acid in their wall. That's the conclusion. Yes, they have mycolic acid. So if the data is red cells, they're acid-fast, they have mycolic acid in their wall. If the cells are blue, the result is they don't have they're not acid-fast, they don't have mycolic acid. With endospores and capsules, it's a little bit vague because your data, you're going to describe the color of the cells. If your cells are just pink, then the result is no endospores. So what's the conclusion? Well, presumably they can't make endospores. They can't produce dormant structures. Capsules the same way. If you see purple cells against a purple background, there's no white halo, they don't make a capsule. I guess you could say capsule is negative, but what it means is they don't store nutrient in a glycocalyx.
Could they have a slime layer that
They might.
isn't stainable?
They might, but there's no way to tell. I mean, because what we're doing specifically is a capsule stain, and usually slime layers don't show. I've seen slime layers pretty good in gram stain, but a lot of times they don't show up that well in capsule stain. Okay, so the other thing is you have to turn in a plate, and that plate has to be a pure culture. You must have isolated colonies. And you have to have a proper label. And the proper label has to include the ID. That culture ID you get by going to NCBI, using your accession number and identify them. So don't turn in a plate that just says morphological unknown number 29. Like, you're going to get docked to have a point for that. I want the name of it, whatever it is. I don't know what 29 is, but look it up.
For the label, that's like name, date,
You have to have name, date, medium type.
but does it have to be in that order?
No.
Is it okay if you
You can
put
put in any order.
Lock it.
Sometimes people write it around the outside edge.
Okay, so that's
It doesn't
fine.
matter where it is
Okay.
or what order it is, as long as it's there.
Okay, good.
But make sure that your label includes the culture name, genus, and specific epithet. Okay. What else? You're turning in 1 page of illustration, gram stain, indirect stain, acid-fast stain, malachite green endospore stain, and capsule stain. Now, when you measure them and you get size, shape, and arrangement, that's the actual size, shape, and arrangement. If you measure them again in an acid-fast stain, they're going to be little. Please don't draw them little. Draw them as you see them in the indirect, because that's really how big they are. And remember, the capsule is outside the cells. Don't draw the cells littler. So make them all the same size. In fact, you can make them the same size and the same shape. You just repeat the illustration 5 times and color them different colors. Indirect stain has a dark background. Capsule stain has a dark background. The rest of them does. Okay? If you need paper, the paper, there's unlined white paper right there. So some of you may be still working on this evening, and that's fine. You can continue to work on that. The soil enrichment, I put the information on the website. That should be pretty doable. The fungus homework, that's just a homework assignment. It's in the lecture syllabus. You go visit the Fifth Kingdom. It's a specific fungus website. The one thing that you have to do is when you get to that website is make sure that there's a place where you click to see full documentation or full information. If you don't, they leave out some categories. If you want to be able to answer all the questions, you have to get the full thing, the whole picture. Okay? So some of you may still be working on this tonight, and that's fine. But we still have to introduce our new groups. So one of the things that you'll notice in lecture, I'm giving you some example genus names, but beyond that, I'm leaving out taxonomy. So in lecture, I'm not talking about domain, kingdom, phylum, class, order, family, genus, etc. Most of that is going to be left out. It's in lab where we start running into the taxonomy, and even there, you don't necessarily get all of it. So, introduction to microscopic algae. Some of this is repetitious from information presented in lecture. Science or study of algae is phycology. The root word phyces means seaweed. So again, people were looking at macroscopic forms initially, and here we see an image of macroscopic algae. Mostly people were interested because they could eat them. They could eat at least some of them. In this class, we're interested in the microscopic forms, and what we know about them, a couple of things. They are oxygenic. They are photoautotrophs. So they produce oxygen as a byproduct of their metabolism, which we'll talk about later on. They use light as their source of energy and inorganic carbon. They mostly contain green chlorophyll pigments, but some of them have other pigments, and they were originally categorized based on pigment. This is talking about them using carbon dioxide from the air. Well, that's what autotroph means, that they can pull carbon, inorganic carbon from the air and use it to make organic compounds, which is then potentially food for other organisms. They are producers at the bottom of food chains and food webs. They were originally categorized based on their pigment type. So here we see multicellular algae, green, red, and brown. When we start talking about the microscopic forms, most of the ones we look at are green. Most of the freshwater algae, I believe, are green algae, and those are the ones we concentrate on. We don't look at any reds. Now, they used to be categorized in the kingdom Plantae, because they were plant-like, being green and oxygenic. But again, we're going to put them in the kingdom Protista. If you look in other books, you will see some algae categorized as Chromista and some categorized as Alveolata, and never mind. We're just going to go with simple. Keep it all the single-celled animal-like and plant-like organisms, we're going to call them Protista. So the kingdom is Protista. And part of the reason I'm doing this is because the taxonomy outside of this is really inconsistent. You pick up 2 or 3 different books and you'll get different categories for these guys. So until that settles down, we're just going with simplistic and saying, we're just going to call them all Protista and not worry too much about it. And the other thing that's somewhat problematic is the fungi are getting mixed in there too. It's like some of the fungi fit into the Protista category. It's very confusing. So what do you have to know? You're going to be able to recognize and name a series of algae. Most of them are green. We have a couple of other examples. You need to know the taxonomy. So the taxonomy is going to be domain. Domain is eukarya. Kingdom. Kingdom is always Protista. And then we have three three phyla, well, three phyla that we have representative slides for. We're gonna skip class, order, family, and jump to genus. So it's very similar to the taxonomy for the fungi. The good news is with the algae, you don't have to know sexual and asexual spores and all of that business. So we only do that with fungi. So here are our examples in a highly repetitious mode. Domain Eukarya, kingdom Protista. Our first phylum is Chlorophyta. What's handy with the fungus phyla, all the fungus phylum ended in Mycota. All the algae phyla end in phyta. Again, phyta meant plant, but it is, at least it's handy. If it has a phyta ending, you know they're algae. Some references call it Phycophyta. We don't need to go there either. Anyway, Chlorophyta. So chloro refers to the green chlorophyll pigments. This is the phylum for green algae. And it is our largest category. We have quite a number of representatives. Most of the ones, I think all of the ones that we look at are freshwater algae, and most of the green algae are freshwater, but some of them live in marine habitats. Some of them even live in damp soils. What's interesting about these guys, once you get into how are they categorized beyond Chlorophyta, people started looking at the shapes of their chloroplasts. Some of them have spiral chloroplasts, some of them have star-shaped chloroplasts. So they kind of identified them that way. It wasn't until quite a bit later that people started categorizing them based on nucleotide sequences, and that's why the taxonomy is all messed up now. So they started doing that. It's like, oh man, these guys aren't related at all the way we thought they were. Anyway, our examples include Chlamydomonas, Cladophora, Aedygonium, Spirogyra, Ulithrix, and a group called Desmids. But desmid, notice how Chlamydomonas, Chlorophyta, Cladophora, Aedygonium, Spirogyra, Ulithrix are italicized. Desmid is not. Desmid is just a common name for a group. It's not a genus name.
A common name for a what?
It's a common name for a group of algae, and I'll explain the characteristics that they have, but they come in a wide variety of shapes, and there are multiple different genera. Okay, so here's our first example. Domain Eukarya, kingdom Protista, phylum Chlorophyta. This is Chlamydomonas. So I mentioned Chlamydomonas in lecture. They're the ones that make the watermelon snow. They can contain phycobilin pigments, red pigments, but typically you see them as green like this. These are very common in situations where people have swimming pools that are turning green. If you have trouble with swimming pools turning green, it's often Chlamydomonas. They also grow in fish tanks and turn the water green. They are little single-celled organisms with two flagella. So here you can see the flagella coming up at one end. They're roughly oval-shaped, maybe a little bit more pear-shaped. The flagella come off their narrow end. On our prepared slides, they're not nearly that nice looking, because on the prepared slides, you can't see their flagella. Well, maybe you can if you have a really good imagination. If you look at the front row, can you see one flagellum here and one flagellum there?
No.
Cool.
Yeah.
Well, they're there. The problem with these things, the Chlamydomonas are prepared so they're fixed, they're flat, but their flagella within the space between the slide and the cover slip can go up or down. So you kind of have to focus up and down to see them. They don't look like much on a prepared slide. They're just these kind of oval to pear-shaped, single-celled algae. You don't need to magnify those with the 100x objective. Probably you can see them just fine with 10x. Don't go higher than 45. Because these are eukaryotic, they're pretty big. Even though that says 1,000, you don't need to go there. The next example, still in the domain Eukarya, kingdom Protista, phylum Chlorophyta. This is the beginning of a group of organisms that are filaments, and this is called Spirogyra. How many people are familiar with the term Spirogyra? It's the name of a jazz band, I think. But it was the name of algae first. Anyway, they get their name for their spiral chloroplast. So here you can see the chloroplast spiraling on the inside of the cell. This one also has a spiral chloroplast, but here the spiral is much tighter, so it's difficult to see. Here the chloroplast spiral is very loose, and there are actually 2 spiral chloroplasts per cell. So there's a lot of variation in Spirogyra. Multiple different species. Most of our prepared slides don't tell you what species is on them, and sometimes they don't because there's more than one. So this particular prepared slide, you see a very large diameter spirogyrotype that has 2 chloroplasts in a loose spiral, and then you have this littler one that has 1 chloroplast in a tighter spiral.
So when you're showing us these prepared slides, are these our actual slides that we
These
have?
are pictures taken from our actual prepared slides.
So this is something we'd actually see, like on a
Yeah.
test. Okay.
Yes. In fact, the images that are on the website are from the slides that I have in a box at home that says test slides. So they look very much like the pictures because they were used for the pictures. And the slide that I have has 2 different types of spirogyra. Well, so if you find these things live, the diameter of the filament that these guys make, it can be as thin as fine silk, or it can be as like the diameter of a horsehair, really coarse. They're just different species. So most of these, we don't know what the species is, and like I said, sometimes there's more than one. The next example is Eulothrix, still in the domain Eukarya, kingdom Protista, phylum Chlorophyta. The characteristic of Eulothrix that kind of gives you the name, it has to do with the shape of the chloroplast. Think of a U shape. You've got a good imagination. Imagine the ends of it bent over, and now it's turned like this. So as you're looking into these cells, you can see the upper surface of each chloroplast. See these here? And then the U shape is down and around, back up. Really, it's like that. Sometimes there's only 1 chloroplast per cell, sometimes there are 2, but they're U-shaped chloroplasts. Our prepared slides look like this. So the color here is not normal color, these are stained, but you can kind of see the end of a chloroplast here and here. The chloroplast goes down and around. If you're familiar with fish vertebrae, that's what those look like. Blue stained fish vertebrae. If you're not familiar with fish vertebrae, never mind.
You said all these slides would be online?
They are on the website. That's why they're up there, so you can look at them. It's a good idea to at least look at the slides in the boxes one time, so that you can, because you need to know, there's variation looking through the microscope. But yes, the images are on the website. Furthermore, when I set up the lab exam, it's my job to find images that look as much like this as possible. I'm not going to be trying to give you something that is off the wall. It's like, here, let's give them this thing they've never seen before. Tell me what this is. That's hardly fair. Okay, the next example is Aedagonium. Here's a spelling, it's just so how it is. So when you see O-E together, it's pronounced eh, which I don't do well. So really these are pronounced Aedagonium, but anyway, it's not Oedogonium, it's Aedagonium. These tend to form mats or clusters, and probably the most characteristic thing that we see on the prepared slides that make them distinct is this structure. This is actually a cell formed as the result of sexual reproduction, so it's a zygote. But they tend to be spherical, and they'll be interspersed with these other cells that are more linear. To me, they kind of look like a watch, if you're looking closely. Or so if you just took this little piece, it's like the face of a watch on a band. It's like, oh, sorry, nobody wears those anymore. Nobody's got their cell phones in. Well, anyway, these are sexual reproductive bodies. There's no particular shape to the chloroplast, and a lot of these you can't even see the chloroplast. So what you were looking for is this series of linear cells and then one fat one. Sometimes they're very spherical, sometimes they're more of a spindle shape, but you see these cells that are distinctly larger than other cells. That's Aedagonium. And then our last example of, this is again a filamentous algae, but it's a branching filament. This is Cladophora. And the Cladophora cells are quite large, probably even 10x is more magnification than you need to see Cladophora. You're going to see these best if you use the 4x objective. So they are microscopic algae, but they're right on the border of being macroscopic. Here you can see a cluster of these things going on the bottom of a fish tank. The cells produce these branching filaments, and they branch and then they branch again. This is the only one we have that's filamentous and branching. So Chlamydomonas was single-celled, Ulithrix, Spirogyra, Aegonium, all filaments, and then Cladophora is branching filaments. The desmids, oh well, this is showing the prepared slide, sorry. Here's our prepared slide of Cladophora. Again, the color is artificial, so they look kind of blue. If they were live, they're green. Some of them have diatoms stuck on them. If you see these funny little things that look like saucers, or actually they look like petri plates stuck on these branching filaments, they're diatoms. So this is the group. As I said, desmid is a group name. It's not a genus name. There are multiple different algae that are desmids, and here you see them, different shapes. What's characteristic about desmids is the cells tend to form a mirror image pair. So this is one cell, and this is another cell, and they're connected right here by a little tiny piece of tissue. Same thing here. When they reproduce, the two pieces come apart, and then they grow a new cell on the inside. So there's two cells, they're called hemicells, half cells, and they're connected by a little piece of tissue in the middle. This is also, but it's hard to see, they're connected right here. So they come in a lot of different shapes. Live ones are some of the most beautiful algae, the green algae. They're really pretty. Our prepared slides of them are kind of strange looking, and it's partly because of the way they're prepared. So what they do with these is they'll collect up some desmids and they stain them pink, and they collect them some different desmids and stain them turquoise, and then they mix them up. So you get some of each. Some of these are kind of triangular, some are kind of star-shaped, some of them look like hippopotamus faces. They come in a lot of different shapes. So those are our desmids. I think that probably 10x is the best magnification to look at desmids. So these, again, these are eukaryotic organisms. They're pretty good size. You don't have to use a lot of magnification. So that's our phylum Chlorophyta, the green algae. The next phylum that you need to know is the phylum Bacillariophyta. Well, if you look at the front of that word, bacilla, that might remind you of bacillus, which is rod-shaped, right? And then remember I said phyta means plant, and besides all the algae phyla end in phyta. So if you can remember bacilli at the front and phyta at the end, and there's a couple of syllables in the middle, you've got Bacillariophyta. This phylum includes the diatoms, and diatom is another group name. So there's a tremendous number of algae in this group, a lot of different genera. All you need to know is the common name, diatoms. What's unique about them, they have these walls made of frustrals, two pieces of glass, like a petri plate, that come together, the two pieces. They reproduce by that shrinking division process. And they come in shapes, so Bacillariophyta, group name diatom. Their shapes, they can be bacilli, linear, they can be triangles, they can be circles. They're always flat, like diatoms are flat, with two frustrals, but the shape of these things is quite variable. The prepared slide that you will see for the exam has 12 types, and one of them looks like a Coke bottle. So they have a wide variety of shapes that these things come in, and different sizes, but remember that's because they do the shrinking division business. But in this image up here, you can see a lot of different shapes. So this is kind of a triangular shaped one, this is a circular one, this is kind of a spindle shape. These are long spindles with these spines off the end. What's kind of interesting about diatoms, the glass walls, the frustrals, have little indentations in them. So they're made of glass, but when you look at them with the microscope, if you focus up and down, you'll see these patterns that appear on the surface. Here you can see the patterns looking like lines. Here they appear to be kind of circles. Here again, lines. Those are made of holes in the glass. So if you look at photoelectron micrographs, they look like that. I try to tell students that image on the right this is a secret language. There are these aliens that came down and they've left us a message in diatomese, but we can't read it. We don't know what the heck they're saying. And if you believe that, yeah, anyway, here again, the colors are completely artificial. So this is somebody having fun with color, with electron photomicrographs. They do that a lot. Yep. Cool images. Anyway, the next phylum, so we're still in the domain Eukarya, kingdom Protista. Now we're going to a phylum that depends on whose book you pick it up. It can have any one of three names. It's called Pyrophyta, or Dinophyta, or Dinoflagellata. The common name for the group is Dinoflagellate. And this little example plush microbe up here kind of shows you the pattern of how these things are put together. The dinoflagellates always have a groove that runs around the center. Sometimes they're just little spherical looking cells. Sometimes they have spines that go one up in one direction, some down in another direction. But this little groove has a flagellum in it. And the flagellum that's in the groove, when it beats, it causes this thing to spin. So the term Dinoflagellata has to do with that spinning motion. Pyro refers to the red color. These guys cause red types, so they have phycobilin proteins or phycobilin pigments, and the red refers to fire. Pyrophyta means they produce the red color. Dino has to do with spinning. Phyta, again, means they're an algae phylum. The Dinoflagellata name is because these things were claimed by zoologists. They thought they were protozoa. They called them Dinoflagellata as opposed to Phyta. I'll accept any one of those names for that phylum. We have two specific examples. This is talking about, again, the central groove with that flagellum in the groove that causes them to spin. If you watch live ones, they constantly are spinning. We have two examples. So this is showing them in a red tide, that image in the middle. This little white thing over here is a boat. So this is a fishing boat on the ocean, and this is the red tide. This is one of our dinoflagellates, and this is the other one, kind of, they look like that. The one on the left is Peridinium, and Peridinium looks a lot like this one, but this is Alexandra. So here's Peridinium on the left, and the one on the right is called Serratia, but it's not spelled the same way. Actually, it's Serratium. So we looked at Serratia marcescens, the red-colored bacteria. It was spelled S-E-R-R. This is called, or spelled C-E-R. Oh, one other thing I should mention, if you look at the image of the dinoflagellate here, you can see this transverse groove that has a flagellum in it. But think of these things as being built kind of like a yo-yo, where you have the top part and the bottom part and a groove with a string in it. If this is turned this way, when you're looking at it on a prepared slide, you can't see that groove. So when you look at a prepared slide, you can see some of the cells will show that groove and others won't. And it has to do with what position they're in. If they're this way, you can't see it. The serratium always has a transverse groove here and a spine going this way, but they can have either two spines or three spines coming down. To me, they kind of look like Eiffel Towers with a leg missing. So, domain Eukarya, kingdom Protista, phylum Pyrophyta, Dinophyta, or Flagellata. Here's Peridinium. Electron photomicrograph on the left, our prepared slide on the right. As soon as I say that, I think some of our prepared slides don't show the Peridinium as just that kind of pink color. They've put some different stain on the wall, and the walls are blue. So you may see them with blue walls. Here's Serratium. Oh, the other thing about the Serratium slide, so there you can see the dinoflagellates, the Serratium, with two spines going one direction, one spine going the other, the central groove in the middle. On that same slide, there's a bunch of other kinds of organisms. There's a bunch of what are called rotifers. They're multicellular organisms, and they're just randomly scattered about on the slide. So if you find things on that slide that don't look like anything you've ever seen before, don't look at it. Find the serratium. They're on there.
But in an exam, you would have the slides
Yes,
positioned
so
with
again,
the pointer.
my job on the exam is make sure that what you're looking at is serration,
Okay.
as opposed to some of the other junk on there. And as I said, dinoflagellates are sometimes categorized as protozoa, which they're not. We talked about them in lecture already. So those are the algae, but because we're doing the entire Protista group, we also have to look at the protozoa. Wait, that's the
I think it's gonna be one.
It's not too bad, but it's super cool.
It's not broken.
Okay, so this is the introduction to protozoa. Again, what makes them different than the algae? Algae are photoautotrophs. Protozoa are chemoheterotrophs. But Euglena, which used to be categorized as algae, aren't. They're protozoa, and they do have green chlorophyll pigment. So most protozoa are chemoheterotrophs. There are some exceptions. They were initially considered animals, so they would have been in the kingdom Animalia, and then within that kingdom there were phyla based on mode of locomotion. So there were ciliates with cilia, flagellates with flagella, things called sarcodyna that had pseudopodia, and then the sporozoans that couldn't move by any means. It turned out that those means of categorizing weren't particularly accurate, but the term sporozoa goes way back in the medical literature, so you will still see that term used. There's a group of protozoa that are called sporozoans, and it had to do with they didn't have any means of locomotion. Well, so now we know that the categories of protozoa have changed, so there are some new categories and things put in groups together in new ways. Again, we're going to just call them all Protista, and we're going to keep this section as simple as possible. So you're going to need to recognize and name them. You need to know the taxonomy as it's applied in this class. All of the fungi phyla end in Mycota. All of the algae phyla end in Phyta. It would be nice if all of the protozoa phyla ended in Zoa. They don't. And there aren't too many that you have to know. So here they are. The first phylum is called Archaeozoa. And that name was applied because these organisms were thought to be ancient forms. And the reason they were thought to be ancient is because they don't have mitochondria. So they thought these must have been an organism type that evolved before prokaryotic cells got into the eukaryotic cells and became mitochondria. New research apparently shows that they had mitochondria and they lost them. But anyway, that's how they got to be called the Archaeozoa. Some other references call them Diplomonadida and Parabasilla. We're not going to. And our examples are Giardia and Trichomonas. So we looked at these in lecture just a little bit. In the domain eukaryote kingdom Protista phylum Archaeozoa, this is Giardia. So this is on the left. Actually, both of those images are electron photomicrographs. That's what you would see if you're looking with an electron microscope, because the flagella are pretty obviously visible. And the other thing is, notice how those things look three-dimensional, like they're really plump. They are not. When you see Giardia live, they are shaped like kites. They're very flat. They swim around, but they're flat. Consequently, if you look at them this way, you can see that teardrop shape. But if they're turned this way, they just look like a little slip of material. They don't have any noticeable shape at all. Now, we're going to look at two different stages. The trophozoite, which looks like this. Again, look for the teardrop shape. The slides, the prepared slides of Giardia, you cannot see that unless you have the 45X objective. They're in stool sample material because they live in the gut. So the slide isn't stained a particular color. These things are just kind of gray or tan. But look for the teardrop shape and two nuclei. So that's what the trophozoite looks like. And again, they have flagella, but we don't have flagellar stains, so you can't see them. The other stage that you're looking for is the cyst. And the cyst just looks like an oval. Some of these ovals, you can see 4 nuclei in there because there are 2 trophozoites per cyst, but most of them you can't see anything. So that's Giardia, not terribly exciting looking. In the same phylum, Archaeozoa, we also have Trichomonas. So Trichomonas vaginalis, the image on the left, the big cells that are in there are skin cells, epithelial cells. So you see the Trichomonas, they're not very big. But on our prepared slides, they are prepared with a flagellar stain, so you can see their flagella very nicely. And they're bright purple. They show up really well. The question on there, do you think they're gastrointestinal parasites with a name like Trichomonas vaginalis? Not so much. So they're sexually transmitted. The next phylum is the phylum Amoebozoa. So these are amoebas, or amoeba-like organisms, and a characteristic of the amoeba is that they have these extensions of the protoplasm called pseudopodia. They have a wide variety of shapes. There are 2 groups that we're going to be looking at, the amoeba and the radiolarians. Some references put those into 2 separate phyla, but we're not going to. So here's our domain Eukarya, kingdom Protista, phylum Amoebozoa. That's what an amoeba looks like. This is an image of Amoeba proteus. If you were looking at live Amoeba proteus, they're almost transparent. The color that you see here, these are food vacuoles. This thing has been eating a variety of materials. There's the nucleus. Here's a contractile vacuole. Here, this is the nucleus. This is the contractile vacuole. This is forming a food vacuole. It's getting ready to eat that little gob of algae. Our prepared slides of amoeba show them looking like that. Most of them are pink, and you can see them in a variety of different shapes. The nucleus is stained a slightly darker pink. However, you should know that I have a prepared slide of amoeba that has green ones and yellow ones and pink ones and brown ones and purple ones. How do they come like that? They're all artificially colored. So somebody took the time to make a batch of amoeba, stain them one color, another batch of amoeba, stain them another color, and they made all these different colors and then put them together. So they're just a mix. That's, again, just to remind you, remember our cheek cells, they were blue, purple, or pink. It was dependent on the stain. That color is artificial. Here we're still in the domain Eukarya, kingdom Protista, phylum Amoebozoa. These are the radiolarians. What you're looking at mostly are glass skeletons. The radiolarians have a body much like an amoeba. They produce a lot of pseudopodia that are really thin, and they extend out through the holes in the skeleton. On our prepared slide, they look like this, these skeletons. It's kind of hard to see. This is not in really nice focus, but all of these, here you can see the holes, but there's holes throughout. These things look a little bit like some kind of architectural structure, like the dome on a building. They're very three-dimensional. So they're like the diatoms in that they're glass, but diatoms are flat. Radiolarians are not. The other thing is about radiolarians, they also lived in water, and their skeletons would sink to the bottom of the water. And so over many years, thousands of years, these things formed also a sedimentary rock type. It's called radiolarian chert. And you can see this at the north end of the Golden Gate Bridge, if you happen to. Well, you can't just stop on the freeway. But there is a park right there that you can go up and you can look at the radiolarian chert. And people have taken samples of this, and then they make really thin sections of it that you can look at with a microscope, and you can see the radiolarians in there. Which is pretty cool. It's mostly glass. So it's right there. The north end of the Golden Gate. Okay, the next phylum is the phylum Apicomplexa. This phylum was apparently named because at the apical end of the cell there was a structure they called some kind of a complex, and they just made this name up. It's like, that's a terrible name. It doesn't end in zoa. How do we know that's a protozoa? Well, I looked at this and I was looking for something that would give us the name Sporozoa, because as I said, sporozoans are medically significant protozoa. So the class is Sporozoea. In the phylum Apicomplexa, class Sporozoea, that's where we find Plasmodium. So this is the sporozoan representation. Plasmodium vivax is the example. This is what, well actually these aren't all Plasmodium vivax. So the Plasmodium organisms, as I said, these are human parasites. They go into human red blood cells and they reproduce inside red blood cells through multiple fission or schizomeny. So you can see different stages of this reproduction inside the blood cells. Plasmodium vivax produces a single ring. So something like this, a ring, and then you can see the development of these schizons. These are eventually going to be the merozoites, and that blood cell is going to rupture and they all get out. When you see multiple rings, that's not vivax, that's one of the other species of Plasmodium. So our slides are Plasmodium vivax. To see this ring, you really have to use the oil immersion objective. So there's blood cells. If you're looking at a blood smear, some of you are familiar with seeing white blood cells, yes? So white blood cells are bigger, usually at least a little bit bigger than the red blood cells, and they have quite a lot of purple color within them. When you're looking for Plasmodium, you're looking inside red blood cells. So these are all red blood cells, and here you see in this one, that ring stage. So that's the ring stage of Plasmodium. Not very impressive. If you pull up a Plasmodium slide and you look all over the place and you don't see anything that looks like that, get a different slide, because we have some slides that are really good and we have some slides that are terrible. Don't spend a lot of time looking at the bad ones. Again, these are vector transmitted. They're all transmitted by Anopheles mosquitoes. The good news for you is you don't have to know Anopheles mosquitoes. We will talk about mosquitoes as vectors, and we look at the Culex mosquito when we get to multicellular parasites. Anyway, inside the mosquito, they reproduce with syngamy. Sexual reproduction happens in the mosquito. Moving on, now we're coming to the domain Eukarya kingdom Protista, phylum Ciliophora. Again, notice the phylum names all don't end in zoa, but these are the ciliates. This is a very large phylum. Most of these are free-living organisms. They're in a wide variety of habitats, most of them freshwater, but a lot of them can live in saltwater, some live inside other organisms. Our example is Paramecium. So, domain Eukarya, kingdom Protista, phylum Ciliophora, Paramecium. There is a study guide that I will give you that is a single sheet, 2 sides of a single sheet, and I'll give you a bunch of species names on there for the different Paramecium. So they come as Paramecium aurelia, Paramecium caudatum, Paramecium ursaria. If I show you a Paramecium, you can use any species name. And for our laboratory exam where you have to identify these organisms, species names are extra credit. You can get up to 5 points extra credit with species names. The reason I put them as extra credit is because the algae don't have any. In our cyanobacteria group, there's only one that has a species name. The good news is, with the protozoa, you may have multiple different species. Now, would I give you multiple points for the same protozoan? I don't know, I'd have to think about that.
When you say that we could get points for knowing the species names, do you mean like you're going to show us one Paramecium and we need to know whether it's Aurelia, Caudatum, or Ursaria?
No.
Okay.
You can use any of them
Oh.
for whatever Paramecium you see. Now, there's another aspect. Do you feel overwhelmed by the Latin that's being thrown at you?
Yes.
Yes.
We have a relatively limited amount of time in here. This is an introductory class. So the other thing that you get when you take Lab Exam 1 is a name sheet.
What?
It's a piece of paper that has domain names, kingdom names, phylum names, genus names. No species.
Oh.
Species are extra credit. And those other names are not in taxonomic order, they're in alphabetical order. But at least you don't have to worry about spelling. So you will have them in your hand. Okay? Yeah, because otherwise, this is a bunch of Latin coming at you very fast.
Oh, I'm freaking out for that. Dude, I was like.
So here's our paramecium. They do have a cytostome. Don't bother to look for it on the prepared slide because you can't see it. The other thing I should point out, that paramecium slide, that's the one on our website. Somewhere in this building, there was a paramecium that looked like that, because I took a picture of it, and I've never seen it since. What's unique about that one is you can actually see the cilia on the surface. Most of our paramecium slides, they look kind of like fat cigars with a label in the middle. That's the nucleus. But the cilia don't show well at all. They're kind of big, kind of spindle-shaped things. They look like that, supposedly. A lot of ours are pink or blue or orange. They come in a variety of shapes. Next phylum is the phylum Euglenozoa. These are flagellated, and some of them look like algae because they have chloroplasts. Most of them are free-living, but what people have found out is that the hemoflagellates, the blood parasites, trypanosomes and leishmania, they're more closely related to Euglena than anything else, so they are in this phylum, the phylum Euglenozoa. So our first example is Euglena, and again, Euglena were initially claimed by botanists as algae because they're green. They're only green sometimes, which is kind of interesting, but they do contain chloroplasts. They also have a little pigment spot at one end that's red. So if you look at live Euglena, you'll see that. Their flagellum comes off the pointy end, and what's interesting about watching Euglena move, they don't push themselves like a tadpole, they pull themselves. If you watch them, the flagellum is at the front, and it's pulling them. You can just barely see the flagellum, like here, on that image. On our prepared slides, you can't see the flagella, but what you'll notice on our prepared slide is that Euglena are not all the same shape. The paramecium always look like that spindle shape because paramecium has a pellicle. It's a kind of a semi-stiff layer on the surface, and it gives them a characteristic shape. These guys have no cell wall, and they don't have a pellicle, and they are very pleomorphic. They change shape. So they can be a sphere, they can be a long, they can be kind of a spindle shape. So you'll see that kind of a slide. They are pink. Actually, there's two images on the website. That one is the better one. The pink with a lot of different shapes. The other one, they all look like kind of blue. They all look the same shape. And that's not realistic. Euglena change shape. They're constantly, as they move, they can stretch out, they can squish up, and they do. And then our other example in the phylum Euglenozoa are trypanosomes. Now these images, the electron photomicrograph, that's where the image for this thing, the reason it's purple is because of that artificial color. These are in the bloodstream, but they're not inside the blood cells, they're between the blood cells. So here you see them. This is an actual, a light microscope image with, these are the blood cells, these are the trypanosomes. They have a dark nucleus that's visible, and they also have that spot at one end, the pigment spot, which Euglena also have. And they have this flagellum that extends along the surface. It's kind of hard to see this, but you might be able to actually see it if you look closely. So here we see a a trypanosome gathering. The trypanosomes have their pigment spot at one end, but notice this kind of wavy surface there. I believe that image was taken with a 100x objective, but you can actually see that kind of fin-like thing with the flagellum enclosed in a double layer of membrane, and then it comes off the end. So those are trypanosomes. Again, they're between the blood cells. Leishmania cells look very much like that. Our slide boxes don't have Leishmania in them. So we get to look at dead preserved ones. We might have some live protozoa. I'll go look. But, so what are you going to do tonight? You can look at prepared slides. At this point, we've gone through cyanobacteria, fungi, and now algae, protozoa. If you want to look at the prepared slides, what I would recommend that you do, kind of work in groups, and each person have a microscope and set up a slide, and then let other people look at it so you can get through a lot of slides quickly. Please use the slides responsibly. They are expensive. Don't one person take out 5 slides and take them to their, set them beside your microscope. That's really selfish. That means somebody else can't look at it. And please don't switch slides between boxes. And if you really aren't finished with the morphological unknown assignment, work on that. A lot of people are upset because they feel like we didn't have time as we were doing the stains. But we don't have a lot of time. The other bad problem we're running into this semester is there's almost no open lab time. And next semester is worse. They're adding yet another section of micro. So the Tuesday, Thursday schedule is gonna be like the Monday, Wednesday schedule. There's no open lab. Except, I don't even know what's gonna happen on Friday. Anyway, so here's, the other thing I should mention about the morphological unknown, you have to turn the whole thing in at once. The page of illustrations, the report form, and the properly streaked plate. I don't wanna get it in pieces and parts. Does it have an absolute hard and fast due date? Nothing does. But people didn't like that when I told you that last week, and so I said, okay, fine. September 28th. I want it in by September 28th. I also want the soil enrichment and the fungus homework, because you should finish those before the exam. They're learning tools. Do whatever you want.
Are you done with lecturing for lab?
I am.